Today, we are taking you into the field, virtually, that is. Welcome to another Heart NSTA Trust lecture series, The Heartbeat. I'm your host, Kevin Baxter. Today's presentation focuses on the art of plant propagation. This is an essential skill for plant care technicians to increase plant numbers. At the end, learners should be able to 1. Understand the need for plant reproduction and growth in agriculture. 2. Understand the two main propagation methods for flowering and non-flowering plants. 3. Perform basic propagation of flowering plants. Let's join Kirk Williams for another engaging presentation.
Thank you, Kevin. Today we are embarking on another in our series of training in agriculture, and we are focusing on plant reproduction. Propagation is one of those words that have been used loosely at times, but today we are looking at what the word propagation means, and then we'll be demonstrating some propagation techniques that are common in agriculture. Today we are defining propagation for you in terms of increasing plant numbers and the methods by which it is done. We are going to be focusing mostly on what it means to propagate growing plants for the industry. As landscapers, as ornamental horticulturists, as crop production specialists, they have to propagate plants so they can have a number of plants growing for their production. And so the word propagation then means to increase your plant number. In agriculture, we refer to propagation in two ways. They're either called sexual propagation, which means we are using the seeds for growing the plants, or we are doing what is called asexual reproduction or asexual propagation, which is using parts of the plant, we call it vegetative parts of the plant, to increase plant number. When we say plant reproduction, we are saying here that especially in growing plants, the seeds or pollens are produced in a sexual form, meaning two gametes coming together, male and female gametes coming together to form that embryo, which is what is used as your propagation material. In asexual propagation, you can take different parts of a plant, namely the leaves, the stem, the root. There are in some cases when flowers are able to pollinate and germinate, they can have flowers inside of it, which has new plants for your growing. And so when you look at how we do propagation in growing plants, if somebody grows 100 seeds, or they sow 100 seeds, and say 75 of that 100 would have germinated, you would have said you have a 75% germination, which then means that when plants are propagated by seeds, it's not saying that you will get 100% in terms of your growth. Same way in your reproduction in vegetative propagation, when somebody says that they are going to be circumposing 50 plants, it doesn't mean those 50 plants will grow, or they bud, say 100 oranges or limes, any of these plants that are done by budding or asexual propagation. It doesn't mean that all of them will take. And so propagation becomes very important in terms of how we do our agricultural production. Why should a student in agriculture learn propagation? He needs to know it, because here is it now that if you are going to be an entrepreneur or somebody who wants to make money from a nursery production, you have to learn the techniques and learn them well. Learning these techniques means that as an agricultural student, you have to try. And so most times you will be saying to yourself, I can't do it. I don't have a green thumb, like most persons would have said it. That it's not that. You have to learn the skill, practice the skill, and you know your grandmother always say, when you practice things, you become perfect at it. And so in terms of your production practices, you are going to learn today some basic things in plant reproduction. Plants in production goes through different phases. And if we start at the seed starts in the ground, it germinates, and it produces its true leaves. If we are going to be transplanting these little seedlings, we call them, you have to wait until the plants would have generated its first true leaves, which are the leaves that has a defined leaf characteristic, which is a stem, at least a leaf stem, and have its veins and veinlets. Now, in terms of your asexual propagation, the plants that are propagated need to have roots that can survive on its own, or the fusion of the cells allowing the plant to survive on its own, like in budding or grafting. Some plants, especially hybrids, do not reproduce true to their parents. And so that is a reason why we say propagation is important. When you have plants that will not produce seeds that are viable or able to germinate, the other aspect of asexual propagation becomes important. Because here is it. If one needs many plants to grow, and the plant cannot produce enough seeds to grow, you have to take part and parcels of that plant and do the necessary propagation practices to allow it to grow. Types of asexual propagation would be included in your cuttings. We're going to be demonstrating that later on. Your layering, we call air layering or ground layering. Sometimes in some plants like bananas, we have to do what's called division. In grafting and budding, sometimes plants like your mangoes that are hybrid, they have to be grafted so you can have those genetic materials being preserved. Of course, for those of us who want to multiply plants that are what we call clean seed, there are some species of plant that suffers from bacterial invasion or viruses, like in banana. You have to take cells of these plants and propagate them in what's called a tissue culture format, in agar solution. These plants then will produce cleaner seed material because they would have been treated with fungicides and virucides and nematicides, all of these chemicals that allow the plant cells to be clean and free of diseases. Now, we are going to be looking at root cuttings as an aspect of plant reproduction too, because there are some plants like your jackfruit and your breadfruit. Jackfruit, yes, produce seeds that are viable, but they take too long. So you want your plant to come into production earlier. You take the root section of those plants, you cut them in your 4-inch pieces, set them in your sand and allow them to have water and adequate heating, and they will produce a new plant. That means the roots will produce the new plants. In breadfruit, our Jamaican breadfruits do not produce seeds that are viable. So if you want breadfruit plants, you have to do what's called root cutting. Now, in ornamental plants, cuttings are very important, and most individuals who run nurseries need to learn all of the techniques and the plants that are able to reproduce by these asexual methods, and stem cutting is one of the major ones that are used in agriculture. So we do types of cuttings, which includes like your stem cuttings, your leaf cuttings, your root cuttings that we have mentioned before. And so when we are going to be using our stem cutting methods, we use small pieces of plants or pieces of stem to reproduce these plants. That's in stem cuttings. We use rooting hormones at times, and we dip these in fungicides that helps the plant to grow or produce its roots in a faster way. In the leaf cutting methods, we use small pieces of leaves to reproduce new plants. And when we say new plants, mean that you need more plants. So the more leaves that you will have set, or the more leaves that you cultivate or propagate, you'll get more plants generated by leaf cutting methods. Herbaceous plants, which are our soft succulents, like your tunas and aloe vera, these plants, they have a number of epidermal cells on their leaf surfaces that are able to generate roots. And once roots are produced, shoots can be produced in plants. The veins, of course, must be cut in most of these plants to allow the plant to send its new shoot. When we look at stem cuttings in general, for most propagation individuals or the student of propagation, there are conditions under which the plant needs in order for it to grow. Like there are some plants that require certain temperature. Another person want to grow your Texas sages and your euphorbia plants. And most plants like these require an amount of heat for the roots to germinate or for the roots to actually produce. And so we recommend that you learn the techniques in your nursery production practices. As students of this class, you have your plants at home. You're going to try your plants. See which one of those plants are able to generate by root cutting. And those can go by your stem cuttings. Those can go by your leaf cutting. But there's a general rule. Most leafy plants that has very thick leaves, like your peperomia, your leaf of life, or your aloe vera, these plants, because of those thick leaves, they are able to generate new shoots. That's one of the rule of thumb. For herbaceous, at least for our perennial hard stem plants, most of them can generate its root by root cutting. Most trees, it's difficult for them to generate roots by just doing your stem cuttings. So in general, we recommend that you do grafting and circumposing and budding on your trees to get them generating new shoots or new plants. In terms of your stem cutting, continuing, we have different methods or ways that you put your materials together. And we say materials. If you're going to be doing your stem cuttings, you need to have material like your media, which is soil, or peat moss, or your vermiculite or perlite material. These materials should be porous and able to hold enough water so these plants can stay alive for a longer period of time. Are we following? Now, thoroughly moisten your container. What's one of the first steps you need to do? Your container should be moistened. You cut your stem cuttings and you put them in the required pot or place. Then you water, cover sometimes with plastic or any material that enables sunlight to pass through. Sometimes you need to put them in an area that is cool. So you have to play around with these plants. A nursery operator understands plants. And when you would have visited a nursery as part of your exercise, the nursery attendant will tell you his or her technique or styles or method. And it's not a secret, but some person observe and develop their secrets in growing plants over a period of time. You too, you need to develop your own secret of propagating plants. And so you will be surprised then that when you go to your grandfather's home or wherever you go in the country, and you'd have seen 2 mangoes on one tree or one, and how is that possible? Or you have seen 2 different plants growing on one tree. How is that possible? Now you understand that in plant reproduction and propagation, all of these techniques make sense, and they are important as a nursery worker. Now, layering is another aspect. I'm not going to spend much time on layering, because these are older techniques that are used in agriculture to grow new plants. But the simple form of air layering is what we know. And I'm going to be demonstrating that later on as we go on. But the whole idea of layering is for you to form roots on a plant that is still alive. You will observe that a plant has very lovely stem. The foliage is lovely. It's growing luscious. And you are saying to yourself, I need that limb or stem to put into a new pot. Your technique that is going to be used now for that is what we call air layering. And you're going to be doing that as a demonstration later on. In divisions, as an aspect of propagation, we have plants like your bulbs or your corms, your rhizome like in ginger, your tubers, your runners, your stolons for those who do landscape like lawn care and lawn establishment. And of course, some we call suckers. These are what we call division, natural ways of getting plant parts being reproduced. And so it's not a secret that if you want to grow banana, you get a banana sucker. Is that so? If you want to grow dasheen, you get a dasheen sucker. Or you can take the corm of that dasheen, cut it in pieces, allow it to form roots, and you have more dasheen plants to grow. If you want to grow ginger, you don't take a whole ginger and put in the ground. You can put that ginger or cut it in pieces. So that's a rhizome of a ginger. And you have all of these plants growing. We'll be looking closer at the different techniques as you learn the skills involved in plant reproduction. You are going to be surprised that one of these days when you become that person who master plant propagation and its technique, you will be a blessed individual going forward. The methods, however, that we choose is dependent on the plant and the experience. I saw a gentleman just recently who says he can circumpose mango. I said to him, well, it's not a conventional way to circumpose mango because the mango needs a taproot system to survive. So if you circumpose that mango and it produces roots, you plant it. In a couple of years, if one of these hurricanes should come around, the plant would not have a taproot system to manage, so it will topple. So yes, you can grow and have roots, but why would you want to put the plant at a disadvantage in having it done that way than doing the right way that we call in agriculture, which is doing what is called grafting. And grafting, we have different methods, and you as students will now learn all of these techniques by researching the method. But the key to plant grafting and budding is the plant itself. We say to you, and you look at those in your notes, that the plants are special, and we only graft and bud plants that are related. So there's no way you can grow an orange plant and put a mango on it. It doesn't work. Because the genes and the genetic makeup of the plants are different. The genome, the whole lineage of the genes and how they're aligned, they are different. So you could not have one plant being grown on the other one unless they are related. The other thing about grafting or budding is the fact that you need to match the cells. And the cells are important in biology, where you would have learned in class from the previous lessons that the vascular cambium of a plant that has the xylem and phloem cells. Xylem is what you know that takes your water, and the phloem is the food stuff. So these cells are moving up and down, taking and transporting plant food material. That cell is found in the bark, just below the bark and the hardwood, that slimy area. Now, if you're going to be doing grafting and budding, your reason for doing this is to match your pieces of plant on that vascular system so it can grow. So you cut your plant as a wedge or a splice or whip and tongue as a method of grafting. You make certain that the scion, which is the root, they call it the rootstock, is the lower section, and the scion is upper section going to be placed on your grafted rootstock. These two plants, either they match each other, meaning that they have the same size, or when you do budding, you make certain that when you cut that plant, the budwood is actually placed exactly on that vascular system where all the energy is, the xylem and phloem cell that has water and food in it. And they are there tightened. I mean, you tie them together, and they stay there, and they fuse. And the secondary and primary cell development will take place, and the plant will grow because they are related to each other. In grafting also, when the scion is smaller than the rootstock, we do cleft grafting. When they are also smaller, we use side or approach or notch grafting or bark inlay. So a number of different methods are out there, but the idea is to place that scion on the vascular system of a plant in different methods. We are looking today also at budding and grafting. And one of our definitions for budding says that it's a form of grafting when a bud is used. And we have three methods or types of budding that are practiced in agriculture. There's what we call patch budding, just a small patch taken out of a plant that is desired, and we place a plant on the vascular system we mentioned before in grafting. In tea budding, we take that bud from a plant that is already bearing, having its characteristics. And also we have what is called chip budding, just a chip, same bud in it, and we place that on the vascular system of a plant. It's very unique in budding and grafting because most individuals who know of oranges and limes, lemons, most from the citrus family, knows that budding and grafting is a technique that is used in producing these oranges. For roses, those are the roses, crotons, budding, patch budding, and chip budding are practiced very often because you need to have more than one species of plant on one individual plant. To have colors sometimes improves the aesthetic nature of the plant. And so the technique is going to be very simple. Most times when we are doing budding and grafting, the technique has to be involved having your tools ready and prepared. And so today what I'm going to be doing before we finish the theory for this content is to look at the tools that are important in performing budding and grafting and in general plant propagation. We have our alcohol that is important for sanitization and sterilization. Our tools that are used in reproduction, just like a doctor's tool, we need to have them sterilized. And so this is what we call a budding knife. Budding knife is a special tool, especially for those who do budding. It has a special curved area and a sharp beveled edge blade. For those children, it should be kept out of children's reach because it's very sharp and it can cause damage. So we normally spray our tools, ensure that they are sprayed and clean. We have another tool that we call our raffia tape, or we call budding tape. This is what that holds the bud or the grafted material in place, right? We have what is called our root hormone for those who are growing plants, are doing propagation by layering that needs roots to be formed, or for those who are doing cuttings that are sometimes difficult to grow roots, you need your rooting powder, which is actually auxin hormone that is in it. Sometimes the limbs of these plants need to be pruned away, so you need your lap shear to hack off those large limbs to give you space for your plants. We are doing a selection of your bud material. This is just a hedge trimmer. Sometimes you need to trim away your hedges for you to get to where the plants are for your propagation. An important tool is your secateur. Some call it roll cut or snap cut, or all different names that are used. These tools must be sterilized also. When they are sterilized, it prevents transfer of microorganisms to a plant that you do not want to be transferred. We are going to be now focusing on the whole aspect of plant reproduction. And the final thing for your circumposing is your foil paper. You need your foil paper to hold the material, which is your media, growing media, onto your plant. Foil paper is very handy. The older generation are used to the plastics. You know, you have a plastic bag or a bread bag to hold your material. But we want to do things faster, so we use our foil paper or foil wrap as a source of keeping those plants in. Now we are heading into the techniques of plant propagation. And we're going to be starting off today with cuttings. As we mentioned before, cuttings is used in parts of the plant, either the root or the stem or the leaves, for propagation, which means you want to generate your roots from these parts of the plant. Now, there are special techniques that are used in cuttings. And for those of us who understand cuttings, we are talking about the node of a plant is important. It's at the node or the joint. The joint or the node is where most plants will generate a new shoot, right? So we normally make certain that we cut just below the joint, not in the joint for part of your stem cutting. Then we cut what is called in the internode as a second cut. Most stem cutting individuals who do growing plant, you want to have at least between 6 and 8 inches of length, especially for hardy woody cuttings. For smaller woody cuttings like your Duranta Gold, you don't need to be that many because there are so many nodes that are on this plant. So if you notice here, 1, 2, 3, 4, 5, so many nodes on it. So you have between 4, 5, 6 inches of space to do your cuttings. So in this case, you're ensuring that you cut first just below your joint. And for those who understand how roots are formed, they are mainly formed at the joint because that's where the bundle of epidermal cells that produces roots are. So when you make your first cutting at the epidermal cell near the joint, that's when you know you have a good cutting. If you should take a stem cutting below or too far from your node, what you're actually doing to the plant is hurting the plant. The plant will have to rot its way back to the joint before it forms its root. In that case, if the plant suffers for a good while before it actually sends its root. So it's good on your part to just go near to that root node and make your first cutting, and you know certain that your plant is able to generate its root right at the joint there. Now, so in stem cuttings, many plants, and this is where your experience comes in now as an individual. You have your bougainvilleas and your hibiscus, your crotons, your so many ornamental, hard, shrubby plants in your nursery that you can generate roots by stem cuttings. When these are cut now, this is when you want to put your rooting hormones on your plant. And so this is where your indole butyric acid comes in, or the auxin hormone. And you want to just dip your stem tip in the hormone. And now the plant is just cut. That means it's very soft. Moisture is there. It absorbs the hormone in its cells. And when you place this in your media for growth, within a couple days, sometimes we say within 3 weeks, most plants will start to generate roots once they're under the right or ideal condition. So we dip them, and we put them in our media. Same thing applies to your bougainvillea and your croton. And this one is your Duranta Gold. So you make your stem cutting small. You remove all of these lower leaves that are there. You don't need these leaves because they are going to be in the media in any case. So this is going to be also dipped in your hormone, right? Dust off the excess, and you place them in your media that is moist and is ready for growing. In propagation, we know that some plants grow very easy, like your herbaceous plants, the succulents. So these plants do not need the rooting hormones because naturally they produce roots so easily by having the ideal condition for growth. We are going to go straight over into another aspect of propagation that we call circumposing. And in circumposing, the same technique applies because we are talking about the vascular system of a plant being able to produce its new growth. And so in this case now, we need our budding knife, or any knife in particular, but then once it's sterilized and without, dirt, things that will invade the plant cells and causing the plants to suffer or have a disease or a condition. Now, hope you are seeing from where you are in terms of this plant. So most time when we are doing circumposing, we look at a plant and we say, oh, this has a very good foliage. I need this part of the plant, or I need this part, or I need this part. So this plant is growing healthy, leaves are very strong. So you want to put on your circumpose section on the area, and you want all of these sections to be alive and well when it is completely formed in root. And so we are going to be cutting the same technique, just like how we do our stem cutting. The roots are going to be formed just at the node. In circumposing, the roots are going to be formed right at the node. So we make a circle around here, and quickly we're going to do this one. And then we make another circle just below, probably a ½ inch or an inch below this one. Then we take this part off, and this plant is very healthy, so the bark slips very easily. And now the bark slips, and we want to remove now the vascular system. If you allow the vascular system to still maintain on this plant, the plant still take moisture from its root system. So we normally destroy this section by scraping the vascular system off. That means the plant does not depend anymore on water coming from the root system or the food coming down and going to other parts of the plant. You're going to be now depending on the media that's going to be put on this plant. You want to place your rooting hormone here, just under the node. That's where the roots are going to be formed. And then you take your media. In this case, we have sphagnum peat moss. Sawdust can be used also. Take a portion. You place it in your foil wrap. And this is where now roots are going to depend on the moisture from this material. So first, we're going to be leaning it just a while there, just like that. And the moisture should be placed right at the node, not below the node, right at the node. And then, foil is easily handled. So we bend it like that, and we put the moisture there, and then we just squeeze in those sections, right? And this plant is now a circumposed plant. You can now do all of these limbs if you need. And for bigger trees, the idea is whenever you water this plant, some of the moisture will actually, go inside there, and it will keep it moist just in case you have drought condition and all that. We are going to go straight into the other aspect, and we are going to look at grafting in the same light of plant reproduction. So in grafting now, what we realize in grafting is that it's a technique that has to be practiced. The skill of the artisan, the skill of the budder or grafting individual is important here. He understands how the plant is grown, so he makes certain that he has a healthy plant, not a plant that is suffering from drought condition or the soil is dried out. These plants will naturally die, at least the bud will die earlier than if a plant is alive and healthy. We have on display here a plant that was grafted probably a couple of months ago. It's a new plant. It's East Indian mango, probably on one of what we call common mango, Jamaican common mangoes, or it could be a number 11. But this plant is what we call the rootstock. So the rootstock just bear the weight of the plant. the scion material, which is actually a bud wood, yes, scion we call it one, is placed on this plant in a particular way. We are going to be demonstrating today one of the techniques that are used. But firstly, we need to tell you or teach you how do you select your budwood. This limb was cut off a Graham mango tree just today, so it's fresh. No sufferation. You realize the limb is as fresh. If you notice the tip of the bud, it shows that it's actually dormant, which means that this bud would have produced a mango last season. So the flower would have come from here and there was a fruit. Now in this case, this becomes a very good budwood, meaning that it has produced already, so it has a genetic potential in it to produce mangoes going forward. Now why do we do grafting in mangoes? If you should allow your mango to stay for a couple of years, plant as a seed, it takes 7-8 years to produce the mango, some 10. But then seeds, based on what we spoke of before, they do not grow true to type. And the term true to type means that all the genetic material in a plant is not produced in its seed. So for you to get a real East Indian, Jamaican, real sweet East Indian mango, you cannot plant that seed. You have to go back to the source. The plant that produces that has a genetic makeup of what you intend. And so what we're going to be doing today is explain quickly how do we do one of our grafting methods. We need our budding knife, sterilized again. We need our raffia tape, and we need our scion material, our budwood. Now, first thing, what you need to understand, and we have explained that in theory, is that the vascular system of the plant becomes important here. The two vascular systems of a plant must match each other. That's grafting. When they match each other, you tie them together and they stay there under the right condition, they will start to produce the secondary cells, and then all the dormant buds that are on this mango tree or the mango bud will start to produce, and then you have growth taking place. So you have this first growth flush here, there's a second growth and a third, and afterward the branches and the plant exponentially starts to get wider each year. Your mango will start to produce the same year, because if this mango, one seed has gone, if you notice, I think there's a flower being formed on this one already. So it means then that once this mango has come out and start a new shoot, the next growing cycle, it will start to produce flower. And once flowers are produced, you know that fruit will be produced. So in this case, I'm going to be putting on I'm going to do what's called wedge grafting. Wedge grafting is very easy. You take the tip of your mango like that, and some of our environmentalists might be saying, oh, you damage the tree is going to die. But the tree has leaves on it same way, so it's not going to die. And trees will always regenerate. What we're going to do is wedge. So we're going to be cutting into our plant, just like this, slice in the middle to expose your vascular system on both sides. We ensure that the scion and the rootstock is the same. And so we ensure, after that happens, we're going to cut this piece of budwood in a wedge form. And if you notice, the knife is very sharp, and it has to be very sharp, and has to be controlled too, right? That's why I say children are not allowed to do these kind of techniques, right? So now the vascular system is exposed. This one is cut there, and you're going to be placing your budwood right into your rootstock. And you ensure that it goes right down, it's firm, right, firm as that. And now both vascular systems are now aligned. What do you do with this now? You need to now tie these together, and that's where the raffia tape comes in. Tape that has no salt or any impurities. It's clean. That means nothing is going to be transferred to this plant. We take it and we wrap this plant, and we normally start from the bottom of it. And the reason for that now is that each layer overlaps each layer. So if water or we have rain or our irrigation system is on, the plant will not have water in that area to really disturb the whole cell development. So we tie this as tight as possible. Okay, you want to make sure that the cells are meeting each other. Some grafters and budders would place another plastic layer over this, like a plastic bag. that plastic bag will now maintain the moisture in your bud. The budwood should not be exposed to too much sun at this stage because you know sun will dry out the material. So within 3 weeks, most times, the dormant buds that are on this scion will now start to pop. They will now come out and they will now produce a mango plant that is now a true type of an East Indian mango that you actually intend on having. This should be kept in a cool environment and ready. Now the other aspect we're going to be looking at, and those students who would have seen these at home now need to just practice the technique. By practicing the technique, you become perfect at it. You want to have mangoes, you want to have East Indian, Julie, Tommy Atkins, Bombay, Graham, Tringram, all of these very good varieties out there. You want to have them in your nursery or in your backyard or in your grandma's backyard. You want to do propagation on them, which is grafting, to make it work. In citrus family, we mentioned before, there's a technique that is called budding. And today, what I'm just showing you is one of those techniques that have resulted in one of these oranges. This is an Artenique with I think it's one of those sour orange rootstock. And so it's an Artenique orange that was budded. So it was a single bud. And if you look at this one, you'll realize here that a union is right at this juncture. Let me take this leaf off. So this plant was budded using what is called chip budding. So chip budding was used to place a bud. So a bud was placed on this sour orange rootstock, and now you have a grown Artenique orange on it. Now, if you intend to grow Artenique oranges by seed, sometimes these Artenique seeds don't want to germinate. And we call those oranges, they are recalcitrant seeds. That means they die easily. Most times when you see oranges grow, it's 1 or 2. 100 orange seeds are thrown somewhere, 1 or 2 might just germinate. So in this case, Artenique is one of those oranges that you need to do budding on for you to have those plants available for your production aspect. I think that is where we come to our final display in plant propagation techniques. You have to practice plant propagation in your nurseries or at home. The techniques are very simple. Talking to somebody who has experience in the area will show you that that is the way that they have grown these plants. You have seen people growing pimento, and you're saying, how do you get pimento to grow that way, to produce that early? There's a technique called approach grafting, where the two cells join together, the vascular system again. And for students of agriculture and propagation, you have to understand what the vascular system means and how to manipulate the vascular system in order for you to grow plants. Growing plants is easy. It's a technique that is widely not used, but by practicing the technique over the time, you master it. You tell somebody about it. You go to a nursery. These individuals will tell you how it is done. There's no secret about these things. You practice them until you become perfect. The exciting world of plant propagation is what we have looked at during our training here today. You need to practice the techniques, whether grafting or budding or circumposing, stem cuttings in different methods. You can visit nurseries where there are individuals who practice these techniques. You learn from individuals by asking how it is done. And so just go out there, do the skill, practice the skill. And as Hart says, skill is power. You get more power. The more you grow the plants, the more you get experience. The more you tell is the more you learn. So go out there and do your best in plant reproduction and growth. Thank you.
Share with us one new thing you learned from this presentation. Send us your answers to our social media pages or WhatsApp at 876-279-7930. You can also check out our YouTube channel to review all the information from the session. I am Kevin Baxter. See you again on the next Heartbeat.